In the world of learning, every concept is a stepping stone to mastery. Welcome to AHS Guide, your complete companion for mastering anatomy, physiology, biochemistry, pathology, and microbiology. Here, we simplify the complex medical topics into clear explanations with visual pictures and aids and exam-focused tips so you learn smarter, not harder. Whether you're preparing for exams or building a strong foundation for your healthcare career, we've got you covered here. Now today we are diving into the fascinating world of lipids, the energy-rich molecule that plays an important role not just as fats in your body. So these lipids are involved in forming cell membranes to act as hormones sometimes, and lipids are vital for survival as well. We'll explore their structure, classification, and biological importance along with some clinical relevance. At the end of this session, you'll be able to understand what lipids are, but also about the essentials of lipids in our life and how their imbalance can lead to diseases. Now, the contents of lipid chemistry are definition of lipids, functions of lipids in the body, classification of lipids along with the subclasses and examples, definition and classification of fatty acids, essential fatty acids, phospholipids and their importance. Now, the first topic, which is definition of lipids, it involves the key points which are origin of term. Lipids comes from a Greek word meaning lipos, which means as fat. The function of lipids in your body is they serve as a chief concentrated energy storage form, that is they work as reservoirs in your body, and any excess form of fat digested is stored in the form of lipids in your body. Now they also play a vital role in cellular structure, that is they form a cell membrane which is phospholipid layer. They also participate in various biochemical functions. Now the nature of lipids are they are heterogeneous groups of compounds which consists of many other groups of compounds and not just one type or homogeneous type of a group. Now it is difficult to define lipids because of the diversity of the compounds or a group of compounds that is present in the lipids. The solubility of lipids is that they are insoluble in water and are soluble only in alcohol and ether. So you can see that the fat does not mix with the water, and it can be dissolved or soluble in alcohol and ether only. The chemical relationship of lipids is they are related to fatty acids and are mostly utilized by living cells. The comparison with other biomolecules, lipids are not similar or are not like polymers, polysaccharides, proteins and nucleic acid, which consists of many number of molecules in it. But the lipids consist very small, mostly of small molecules. Coming to the functions of lipids, lipids are having several important functions. Four of them are given here. So they are concentrated fuel reserve of the body in the form of triacyl glycerols. So whatever excess amount of fat that is digested in your body is being stored as triacyl glycerols and is used whenever required. When the body is in demand of energy, the fat is broken down into glucose and is utilized by the cells of the body. The second point, lipids are constituents of membrane structures and regulate the membrane permeability. So here is an image of a cell membrane wherein you can see the two layers which consist of a phosphate and lipid groups together forming a phosphobilipid layer which protects the cell membrane from the external and internal environment and allows the certain nutrients to pass through the cell membrane. So that is the role of lipids in cell membrane. The third function of a lipid is it can solubilize or break down vitamin A, D, E and K and provide nutrition to the body. Lipids are also important in cellular metabolic regulators such as steroids, hormones and prostaglandins are also being regulated and controlled by the lipids, or in the functioning of the lipids is involved in steroid, hormone and prostaglandin synthesis. Now the classification, will come back to the image later. Classification of lipids are of three types, which is simple, compound and derived. Simple, compound and derived lipids have subtypes in it which we will be learning individually. Now, simple lipids, as the term says, it is not very complex or does not consist many number of groups in it. So, simple lipids has esters of fatty acids along with alcohols. So, esters of fatty acids plus alcohols make up a simple lipid. So, simple lipid can be further divided into two types. One is fats and oils. The other one is waxes. So, fats and oils are esters of fatty acids with glycerol. Fats are solid at room temperature, oils are liquid. Now fats and waxes have a difference. We'll be seeing after what waxes is defined as. So waxes are esters of long chains of fatty acids with alcohols other than glycerol. So it is not glycerol, not just glycerol that is present in the waxes, alcohol is present in spite of glycerol. So this alcohol that is added in waxes is either aliphatic or alicyclic. Aliphatic is long chains of alcohol, hydrocarbons along with alcohol, or a cyclical structure of a compound. So these main difference between fats, oils and waxes is that fats and oils have glycerol as a component and waxes have alcohols as the component. Now the uses of the simple lipids is they can be used in candles, lubricants, cosmetics, ointments and polishes. The complex lipids or compound lipids, the definition is the esters of fatty acids with alcohols plus additional groups like phosphate, carbohydrate, protein, etc. are added. So for a fatty acid ester along with alcohol and additional groups such as phosphate, carbohydrates and proteins, they form a complex compound. So you can see there are many components inside a complex compound. There are simple lipids. We had only esters of fatty acid plus alcohol, but here we have esters of fatty acids with alcohol with an additional group which can be phosphate or carbohydrate. Not all of them at once, but can be in phosphate or carbohydrate or a protein. Now we'll see the first subtype of complex lipid. Complex lipids first subtype is phospholipid. Phospholipid, as the name says, it has a phosphate group in it. So phospholipids contain a phosphoric acid along with a nitrogenous base and alcohol plus fatty acids. So these are the four components of a phospholipid: phosphoric acid, nitrogenous base, alcohol and fatty acids. You'll remember those four and write the same in the exam as well. Now phospholipids can be further divided into two types: glycerophospholipids and sphingophospholipids. Glycerophospholipids contain glycerol in spite of alcohol over there. Sphingophospholipid as a sphingosine as a alcohol. So what are the contents of glycerophospholipids? It's phosphoric acid, nitrogenous base, glycerol and fatty acid. Together they form a glycerophospholipid. Examples of glycerophospholipid are lecithin and cephalin. The second subtype of phospholipid is sphingophospholipid, wherein sphingosine is the alcohol. Here, the contents of sphingophospholipid are phosphoric acid, nitrogenous base, alcohol, which is sphingosine, plus fatty acids. Together make a sphingophospholipid. So, the examples of sphingophospholipid is sphingomyelin. The second subtype of complex lipids are glycolipid, composed of fatty acids. As glyco is saying, it's carbohydrate derivative. So composed of fatty acid plus carbohydrate plus nitrogenous base. Sphingosine is the alcohol that is present in the glycolipids. This sphingosine alcohol when present in glycolipids is called glycosphingolipids. Simple. Whenever a sphingosine as an alcohol is present in glycolipids, it is called glycosphingolipids. And the examples are cerebrosides and gangliosides. Moving to the third subtype of complex lipids, which is lipoproteins. It has, as the name says, protein. Macromolecular complexes of lipids along with proteins make up the lipoproteins. The fourth subtype of complex lipids are these include sulfolipids, aminolipids and lipopolysaccharides. As the name says, sulfur containing sulfuric acid, amino containing amino acid groups, lipopolysaccharides containing polysaccharide groups. The third type is the derived lipids. So as the word says, derived is being taken from or taken after hydrolysis of simple or complex lipids. So simple and complex lipids are hydrolyzed, that is diluted, and whatever compound or lipid that is derived from that is called a derived lipid. Glycerol, fatty acids with monodiacylglycerols, fat-soluble vitamins, steroid hormones are all derivatives of lipids. They're not exactly lipids, but they are derived from the lipids after hydrolysis. Hydrocarbons, ketone bodies, these are all the derived lipids. So the miscellaneous lipids are the compounds which look similar and function similarly like lipids are called as miscellaneous lipids or lipid-like characteristics. Some of the examples of miscellaneous lipids are carotenoids, squalene, hydrocarbon, terpenes. Now carotenoids, we have heard it in carrots and various other vegetables which are orange color. They are similar to fat-like because they are best soluble with the fats. So that hence carotenoids mimic to be lipid-like. Squalene. The last type of lipids is a neutral lipids. So as the name says, it is an uncharged lipid and there is no free electron that is roaming and is involved in any bond formation. So these neutral lipids examples are monoditriacylglycerols, cholesterols and polyester esters. Now the second topic of the lipid chemistry is fatty acid. Definition of fatty acids are carboxylic acids with the hydrocarbon side chain. So these hydrocarbon side chains are present on the fatty acids along with COOH group or carboxyl groups. These are the simplest forms of lipids and they do not contain any complex additions or branches that are observed. Now, how is the fatty acid obtained? It is obtained or found mainly with the esterified form of lipids and they are also occur as a free fatty acid or unesterified acid. The animal origin are simpler structures, whereas plant origins may contain epoxy ketohydroxyl groups or cyclopentane rings that are attached to the fatty acid chain. Now, there are different types of fatty acids based upon the number of carbon atoms or based upon the saturation or unsaturation, that is presence of double or triple bonds. So the third point of fatty acids, even and odd carbon fatty acids. So even carbon fatty acids, as the name says, even number of carbon atoms present in the fatty acid group or the carbon chain. You can see, for example, it might have 14 carbons or 20 carbons. 16 carbons, 18 carbons. So the even number of carbons present in the carbon fatty acid chain is called even carbon fatty acids, and it helps in during biosynthesis or the breakdown of the carbon. It is a 2 carbon addition step and hence the name even carbon fatty acids. So some of the examples of even carbon fatty acids are palmitic acid and stearic acid, which has 16 and 18 carbons respectively. Now coming back to odd carbon fatty acids, here the carbon chain might have 3 carbons or 5 carbons in the fatty acid groups. So some of the examples are propionic acid and valeric acid. Moving to the fourth key point of fatty acids is saturated versus unsaturated fatty acids. Now saturated are the ones which can be simplified and have single bonds in them. So there is no double bonds present between the two fatty acids. So because of the single bond, the breaking down of the fatty acid is simpler and is saturated. In an unsaturated fatty acids, there are one or more than one double bonds present in the fatty acids and they are usually mono unsaturated and polyunsaturated types. In mono unsaturated, there is, as the name says, one or one double bond. Polyunsaturated or PUFA fatty acids, these are 2 or more double bonds. We might have heard about PUFA in advertisements as well because they contain omega-3 or omega-6 which are found in the marine or fish oils like salmon oil. So these might contain PUFA or the fatty acids which are naturally occurring. So it might be in sunflower oil also. So these are the examples of PUFA. Both the saturated and unsaturated fatty acids are naturally occurring lipids. So how is the fatty acids named? So there are two methods of naming it. One is systemic naming and the other one is common naming. Under systemic naming, it can be in two types again, saturated and unsaturated. In the saturated systemic naming, it ends with anoic. For example, octanoic acid. Based upon the numbers of carbon atoms present, we are going to name whether it is saturated or unsaturated. So in saturated, we name with anoic, which is actinoic. Unsaturated, it ends with enoic or octadecenoic acid. So common names are widely used and commonly used among people. For example, like palmitic acid, oleic acid, etc. Now, carbon numbering. We saw the nomenclature. So, before nomenclature, we must know how to number the carbons. Now, carbon numbering happens when the carboxyl carbon is always numbered 1. So, here you can see an image wherein COOH is numbered as the first carbon atom of the chain. So, you are going to start the numbering from the COOH or carboxylic acid group, carboxylic carbon as the first number, and then other conventions can be used such as Greek letters like alpha, beta and gamma can be used in numbering the branches, and omega carbon is the terminal carbon methyl group. So you can see there is COOH at this end and there is CH3, which is methane or methyl group present in the end of the chain. So omega is used as the terminal or the end methyl group naming convention. From omega end, omega 1, omega 2, omega 3, omega 4 and omega 5, omega 6. So that's how the conventions are being used. Sometimes the example is being given, so you can see the example and understand the way the carbon is numbered in a fatty acid chain. Now the chain length can be classified as short, medium and long chain. Now short chain has less than 6 carbon atoms and medium chain has 8 to 14 carbon atoms and long chain has 16 to 24 carbons present in the chain. So that is how chain length of the fatty acid can be determined. The shorthand notation that is being used while naming a fatty acid, there are two methods. For example, if the CD is a component, CD is the format used to represent the positions of C and D will be number of carbon atoms present is the C and D is the number of double bonds present in the carbon atom. Position is the double bonds position from the carboxylic end is determined. For example, if a palmitic acid is given, so the palmitic acid may contain 16 number of carbon atoms with 0 double bonds. So there are no double bonds in palmitic acid. So this is how we are going to represent or a shorthand notation for a lipids to be fatty acid to be told. Now palmitic acid has 16 carbon atoms with 0 double bonds. Oleic acid has 18 carbon atoms and 1 to 9 double bonds might be present. Arachidonic acid might have 20 carbon atoms. So arachidonic acid has 20 carbon atoms present in it. It might have 4 or 5 or 8 or 11 or 14 double bonds. Okay, so that is how the format of representation of shorthand notation is seen. For a double bond notation, you can have a delta 9 symbol. So delta here double bond between carbon atom 9 and 10 from carboxylic end. Omega 9 is used to represent the same bond but counted from omega end. So in previous wordings we saw this omega, right? So from terminal methyl group, omega is labeled as omega 1, 2, 3 for the carbon. From carbon 7, 6, 5, 4, 3, 2 and 1. So here the omega 9 is the ninth omega terminal carbon atom that is present and that is being labeled at which region the double bond is present. For example, if the carbon atom has the double bond between 6 and 7, you can label it as delta 6 or omega 6 based upon the region of the where double bond is present. Now, the series of unsaturated fatty acids can also be given a shorthand notation, omega 3 linoleic acid, wherein 18:3, delta 9, 12, 15. So here omega 3 is the third terminal carbon atom, and in linoleic acid 18 is the number of carbon atoms present, and 3 is the number of at the place where number of double bonds that are present. So whereas these 3 double bonds present, they are present at delta 9, 12 and 15, at 9th carbon, 12th carbon and 15th carbon. So the terminal carbon is, so there is 18 carbon atoms that is present, along with 3 double bonds. The 3 double bonds are placed at 9, carbon 12, carbon atom 12 and 15th carbon. So the next example we can see there is omega 6, wherein there is a double bond at 6th carbon position. So linolenic acid has 18 carbons with 2 double bonds. The 2 double bonds are placed at 9th and 12th. Omega 9 is the count from the terminal carbon to the COOH group. So there is 9 terminal carbons, and oleic acid has 18 carbon atoms, 1 double bond, and the 1 double bond is placed at 9th. So this is quite important to understand. Now arachidonic acid also I have mentioned over here, wherein there are 20 carbon atoms with 4 double bonds. The 4 double bonds are placed at 5th carbon, 8th carbon, 12th and 14th carbon. So if you want me to explain it again, here is a short form of the notation of a fatty acid. There are 4 methods of representation of a lipids in a short form. So for the format, C and D is being used for an example. So where C is the number of carbon atoms and D is the number of double bonds. So this position is also being placed where after a semicolon. Okay, so for example, here in palmitic acid there is 16 carbons with no double bonds, 0 double bonds. In oleic acid there are 18 carbon atoms, 1 double bond, and the 1 double bond is placed at 9th carbon. Understood? Now arachidonic acid has 20 carbons, 4 double bonds, and the 4 double bonds are at 5th, 8th, 11th and 14th position. Now double bond notation. Here where the double bond is present, the position of double bond is represented over here. Delta 9 can be used or omega 9 can be used. Delta 9 is double bond that is present between carbon 9 and 10 from the carboxylic end. Remember, from the carboxylic end, which is, so for example here we have the image, right? So COOH from here 1, 2, 3, 4, 5, 6, 7 and till 9th group, for example I told. So till the 9th carbon atom, after the 9th carbon atom, between 9 and 10 the double bond is present. Similarly, there is another way to represent double bond, which is omega 9 over here. So omega 9 is where same bonds are bound from between the last methyl group, that is you start counting from here. So omega 1, omega 2, omega 3, 4, 5, 6, 7, 8 and 9. So at the 9th position the double bond is present. Now series of unsaturated fatty acids. So how are you going to name a unsaturated fatty acid? With a shorthand notation. So omega 3, as I told, you're going to count from the terminal carbon. So there are the third terminal carbon, linolenic acid. So there is 18 carbon atoms in linolenic acid and 3 double bonds. The positions of 3 double bonds are at 9th, 12th and 15th. Similarly, the second example, omega 6, from there is the 6 terminal carbon atoms, and linoleic acid has 18 carbon atoms, 2 double bonds at position 9 and 12. Arachidonic acid has 20 carbon atoms with 4 double bonds and at the place of 5, 8, 11, 14. And the last example, which is oleic acid, there are 9 terminal carbons and there are 18 carbon atoms with 1 double bond. The 1 double bond is present at 9th position carbon. Okay, so that is how shorthand notation is. If you didn't understand, rewind the video and try adding certain examples to the fatty acids and start working on the shorthand notation. So the next topic, which is essential fatty acids, as the name says, essential, or which is required and is not synthesized in your body. So it has to be obtained in the nutrition form. So essential fatty acids, the definition, EFA, fatty acids not synthesized by the body and must be obtained from the diet. Some of the examples of essential fatty acids are linoleic acids, linolenic acids, arachidonic acids, and if the linoleic acid is deficient, arachidonic acid can act as a supplementation for it. Now, biochemical basis for essentiality. Humans are not having the capability to synthesize or produce enzymes in order to build essential fatty acids, because these essential fatty acids might contain 9 carbons to 10 carbon chain fatty acids with double bonds. So with double bonds, and synthesizing this is impossible for humans, and hence the essential fatty acid is required. The functions of essential fatty acids are they maintain the structural and functional integrity of cell membranes. That is, they keep the cell membrane in a strong and structure to keep the shape of the structure of the cell membrane, and the functionality, which is the allowing the external nutrition to come into the cell. So that is how it is essential fatty acid is essential. Transport of cholesterol. So it also helps in the transportation of cholesterol. It acts as a transport medium. The formation of lipoproteins. So we saw a type of lipids which are with lipids and proteins together, they form lipoproteins. So the formation of lipoproteins is facilitated by EFA. They also prevent fatty liver. So the unsaturated fatty acids, fats that we consume, might cause fatty liver or the liver cirrhosis. So those can be prevented by consumption of essential fatty acids. Precursors for eicosanoids, prostaglandins, thromboxanes, and leukotrienes. So these are the essential human certain components that are secreted from the human body. So they act as the precursors or the first steps for the synthesis of eicosanoids, which are like prostaglandins, the pain regulating hormones, and thromboxanes for the clotting of the blood, and leukotrienes as well. Now, the deficiency of essential fatty acids may lead to paranoderma, which is the toad skin type of skin wherein you have a patchy skin, and horny eruptions on limbs, back, and buttocks, wherein there are extra growth of the skin, which can lead to hardening of the limbs, back, buttocks, the layer is formed. And loss of hair can also be seen if essential fatty acids are not provided essentially. And poor wound healing, because as we saw here, thromboxane, it is a precursor or the beginner of the clotting factors, is controlled by essential fatty acids. So The next part is isomerism in unsaturated fatty acids. Isomerism is a similarly looking kind of a compound. Now the isomerism can be in geometric isomerism. So geometric is the orientation and the double bonds that is present in the compound might overlap one another. Now there are two types of geometric isomerism, which is cis trans configuration and trans configuration. The cis configuration have groups on same sides, for example oleic acids. So you can see there are groups that are present on the same side. The pink color representation of chemical compounds, which is carboxylic acid and methane groups of carbon chains, are present on the same side. In case of trans configuration, the compounds are present on the opposite sides, which are on opposite sides of the carbon chain or the unsaturated fatty acid. Now cis forms creates a bend or mix. There is increased membrane fluidity. So since the groups are on the same side, the weightage is on the same side, they allow in bending and kinking of the configuration. So these compounds have one side of weightage of the all the molecules and help in restructuring of the carbon atom and also help in membrane fluidity. They don't form the rigidity, whereas they also have, they can form more double bonds and there are more bends also possible. For example, for a cis trans isomer is an arachidonic acid and it can form a U shape. So that is the membrane fluidity that can be seen in case of cis form. In trans form, they are more stable because the weightage is distributed on both the sides. The compounds, the chains distributed on both sides are going to cause the weightage to be distributed equally, and they are more stable and they don't have to restructure. They are usually straight chains and are found mostly in processed foods. The special types of fatty acids are hydroxy fatty acids, cyclic fatty acids, and eicosanoids. The hydroxy fatty acids contain a hydroxyl group, which is OH group, and some examples of hydroxy fatty acids are beta hydroxybutyric acid, a ketone body is also present, cerebronic acid, racinoleic acid are some of the examples of hydroxy fatty acids. The cyclic fatty acids might have a cyclic structures of carbon atoms present in them. Usually they are rare, and some of the examples are kaumogra oil, which is used for leprosy treatment. You can search for the acid and the oil that is synthesized from the fatty acids. Eicosanoids are derived from eicosanoid, derived from eicosapolenoic fatty acids. These include prostaglandins, prostacyclins, thromboxanes, leukotrienes. So these are the enzymes that are released in the human body, and these eicosanoids act as a special type of fatty acids that are released in your body. Prostaglandins are involved in the pain and the flight and hormones, and prostacyclins are kind of fatty acids, and thromboxanes, as I told, is involved in the blood clotting. Leukotrienes are also a part of component of a blood plasma. Triacyl glycerols. So the definition of triacyl glycerols are they are esters of glycerols with three fatty acids. They have the glycerols in the fatty acid groups. So tri meaning three fatty acids with esters of glycerols present in them. These are widely found in the plants and animals as either fats or oils. They are usually insoluble in water and non-polar. So non-polar is they do not react or they are neutral fats. The function of triacyl glycerols is they act as a energy or a fuel reservoir. So they are mainly involved in the fat storage. So the fat storage in human consists about approximately 20% of body weight in men and approximately 25% for women. The energy reserve that is stored is sufficient for 2 to 3 months, and the stored adipose tissue is in the form of globules or in the adipocytes. So adipocytes are the fat tissues that are present, that is a type of a connective tissue that are present in the adipose tissues, are present having the globules of fats that is stored in the fat cells. These are not the structural component of cell membrane and are mainly involved in the cushioning effect of the human body. And it also serves as a energy or a fuel reservoir. Now the types of acylglycerols are monoacylglycerols, diacylglycerols, triacylglycerols, simple TAGs, mixed TAGs, and sometimes they also are in plants as triacylglycerols. Now monoacylglycerols, as the name says, it has one fatty acid, di has two and tri has three. So triacylglycerols are more important because they are seen in human as a storage form as well as a protective form for the human body. Now simple TAGs have three fatty acids similar to triacylglycerols, but they are not as complex and are simple. Examples for it is tristearine. Mixed TAGs have 2 to 3 different fatty acids that are together, two different forms or 3 different forms of fatty acid. So one carbon might be saturated, the second carbon can be double bonded, unsaturated, and third type can be any of them, either saturated or unsaturated. Also, all these 3 carbons are in the same TAG. So these are the mixed TAGs. The primary difference between simple and mixed is the simple has all the fatty acids having same group of fatty acids, but mixed has one saturated, one unsaturated. The other can be saturated or unsaturated. Now plants also have triacylglycerols, which are higher content of unsaturated fatty acid, that is double bonded fatty acids are more in plants compared to animals. So stereospecific numbering is a system where the number of carbon atoms in a triacylglycerol, the glycerol component present in the lipids. So you can number the carbon atoms present in the TCGs. So how is it done? It's usually done by naming the carbon atoms C1 and C3 of glycerol, which are not identical to one another. And the top carbon, there is 3 carbons present. So there will be S1, S2, S3 named for the first, second and the third carbons. So these carbons will have OH groups attached to them. And these OH groups are named based upon the presence of saturated, unsaturated fatty acid or a phosphate group present. For example, here you can see a triacyl groups. So there are 3 carbon atoms present in triacylglycerols. So CH2O, there is one saturated carbon, and here there is second saturated OH group attached to it. The third C double bond unsaturated group. So these are the 3 carbon atoms with different stereospecificity. So stereospecificity is based upon the Fisher projection of the carbon atoms present here. So one of the examples that is present here is glycerokinase phosphorylates. So the stereospecific numbering for glycerokinase phosphorylate is SN3 position, because the stereospecific numbering of glycerol is having the third position with phosphate. Okay. Now the properties of TAG or triacylglycerols are, they are involved in hydrolysis and as well as saponification. Now for hydrolysis, the enzymatic hydrolysis of lipases will give you free fatty acids plus glycerol. So if the lipases are hydrolyzed, the free fatty acids plus the glycerol is present, which is involved in digestion and fat mobilization as well. So hydrolysis is helpful in digestion and the fat mobilization of a compound. Now saponification, the alkaline hydrolysis, that is glycerol plus soaps, will give you an alkaline hydrolysis and leading to saponification. Now rancidity is another type of a property for TAGs. So rancidity is the deterioration of fats or oils, the unpleasant taste and the smell of the oils that you get after opening an expired chips bag. You can feel the smell of the oil that is rotten or is unpleasant. So that is the rancidity. That is because of rancidity. So rancidity can be of two types. It is hydrolytic rancidity and oxidative rancidity. Hydrolytic rancidity is due to bacterial enzyme hydrolysis. Oxidative rancidity is due to oxidation of unsaturated fatty acids. That is, the double bonds will start producing aldehydes or ketone groups or any other chemical components due to presence of oxygen or addition of the breaking down of double bond in a fatty acid. So those are the two types of rancidity. Now how to prevent this rancidity is by adding antioxidants, which thereby does not allow the breaking down of the unsaturated fatty acid and leading to rancidity. So there are two types of antioxidants. One is natural, the other one is synthetic. Naturally, vitamin E or tocopherols, hydroquinone or gallic acid can be used as a antioxidant. Sometimes synthetic antioxidants are also used in food processing units, which is BHA, BHT, propyl gallate. Lipid peroxidation in BIPO. So the oxidation leads to formation, excessive oxidation leads to formation of peroxidases and free radicals, which can damage tissues, leading to age, cancer and atherosclerosis. So the body's defense mechanism like vitamin E, urate and superoxide dismutase are also used as a oxidative agents. So whenever a peroxidation is used, the free radicals are released. Whenever free radicals are released, these are going to damage the tissues. Excessive oxidation leads to peroxidases and free radicals. These free radicals and peroxidases can damage tissue, and these damaged tissue can cause aging, cancer and atherosclerosis, which is formation of fatty blocks in the heart. Body's defense mechanism tries to stop the peroxidation by use of vitamin E, urate or superoxide dismutase. Now the test for purity of fats or oils is the iodine number. So the grams of iodine that can be absorbed by 100 grams of fat indicates the level of unsaturation that is present. So high iodine number shows that more unsaturation, low iodine number shows less unsaturation. So the more unsaturated it is, the difficult. The saponification number is the second type of test that is used for detecting the purity of fatty acids. For saponification, milligram of potassium hydroxide is mixed with the 1 gram of fats and observed for the molecular weight of the fatty acids. If the molecular weight of fatty acids is high, the short chains of fatty acid is observed. For example, in a human fat, there is 195 to 200 molecular weight, and for butter 230 to 240, and coconut oil 250 to 260 is the molecular weight of fatty acids present in the gram of whatever type of fat that is being used for test. Now Richter missile RM number, here an ml of 0.1 normal potassium hydroxide is used to neutralize the volatile fatty acid from 5 gram fatty acids. So you're going to take a neutral volatile fatty acid from the 5 gram fat and neutralize or dilute with the 0.1 normal potassium hydroxide and is tested with the butter's purity, which is approximately 25 to 30, and The other types of fats, they have less than 1. So that is how the butter purity is determined. Richer meal. Now acid number is another method to determine the purity of fats or oils. So a milligram of potassium hydroxide is neutralized with the free fatty acid, which is extracted from 1 gram of fat, and the high acid number shows that there is decomposition or contamination in the fatty acid. So the last topic, which is phospholipids. These phospholipids contain both phosphoric acid and fatty acid along with nitrogenous base and alcohol. So there are two mains of phospholipids which we have already seen: glycerophospholipids, phosphoglycerides, which contain glycerol, and sphingophospholipids, sphingomyelins, which contain sphingosine as the alcohol. So glycerophospholipids are major lipids in the biological membrane, that is the cell membrane. Their structure is glycero 3 phosphate with esterified at C1 carbon is saturated fatty acid, C2 has a unsaturated fatty acid, and C3 has a phosphoric acid plus nitrogenous base. So it is a three carbon atom, wherein the first carbon is usually saturated fatty acid, second has a unsaturated fatty acid, and the third one has a phosphoric acid plus nitrogenous base. Now the important types of phospholipids are phosphatidic acid, where the simplest phospholipid, and it forms an intermediate between triacyl glycerols and phospholipid synthesis. So whenever a synthesis of TAG and phospholipid is happening, an intermediate product that is obtained is phosphatidic acid. Lecithins or phosphatidyl choline is most abundant in membranes, and it is a storage form of choline. So dipalmitoyl lecithin, lung surfactant, is also a type of a lecithin, and deficiency of dipalmitoyl lecithin can lead to respiratory distress syndrome in infants. So if there is no lung surfactant, the baby that is breathing and taking in oxygen, the lung cannot expand and contract properly without the surfactants. So when the expansion and contraction of the lungs or the inspiration and respiration facilitated by these surfactants are not present, it might lead to respiratory distress, that is the baby is not able to breathe. Lysolecithin is formed by removal of a fatty acid from the lecithin. So hydrolysis of a fatty acid will give you lecithin. Now cephalins are another type of phospholipids, phosphatidyl ethanol amine. It contains ethanol amine as a base. And the fourth type of phospholipid is phosphatidyl inositol. It contains myoinositol involved in hormone action, like IP3, DAG are involved in the hormone action. Phosphatidyl serine contains serine, also threonine in some tissues. Plasmalogens, these have ether linkage at C1 instead of the ester linkage, and the major type of plasmalogens are phosphatidyl ethanol amine. Cardiolipins, they are found in inner mitochondrial membrane and are essential for ETC function, that is electron transport chain function. And antigenic properties are deficiency leads to mitochondrial dysfunction and birth syndrome. So it is antigenic in property, that is provides a antigen antibody interaction and leading to killing of any other microorganisms present. So these deficiency of cardiolipin can lead to mitochondrial dysfunction or Barth syndrome. So here are the structures of the different phospholipids here: phosphatidic acid, lecithin, cephalin, and phosphatidyl inositol, and phosphatidyl serine and plasmalogens. Pause the video, see the structures, different structures, and the difference between each phospholipid and the chains, the pink color additional groups that are present along with the phospholipid chain. So here the black color chains that you see are the fatty acids, and the blue color are the phosphate groups or the phosphoric acid, and the pink color is the representation of additional groups that is added. So you can see in lecithin there is choline group, that is the additional group, ethanol amine in cephalin, in myo-inositol in phosphatidyl inositol, and serine group in phosphatidyl serine, and ethanol amine in plasmalogens. Similarly, the cardiolipin, there is an ester chain along with the phosphorus group. We have sphingomyelin also, the 8th type of phospholipid, whereas sphingosine is the alcohol that is present, and ceramide is also present. There is a choline group also attached to the phosphate group, phosphoric acid. So moving to the second type of phospholipid, which is sphingomyelins. Sphingophospholipid contains sphingosine as the amino group along with alcohol, not the glycerol group. So the structure of sphingomyelins are sphingosine plus fatty acid, which gives a ceramide with a amide linkage. The alcohol group is linked to the phosphoryl choline. And these sphingomyelins are present in the myelin sheath of a bacteria, the outer protective covering of a bacteria. In brains and in nerves also, they act as a protective layers. The ceramide function in the sphingosine is they act as a secondary messengers, that is in cell death and cell cycle. They are involved as a second messengers. They are important in skin water permeability as well. So it allows, sometimes skin is percolent, or it allows the water to seep into it. So here the structure. So you can see the ceramide is formed because of the presence of a fatty acid, the black color fatty acid group, along with the sphingosine that is attached to the fatty acid group, which makes the whole black chain is called the ceramide. Along with the addition of phosphate group and a choline group, it forms a sphingomyelin. The functions of phospholipids in general, when you take in consideration of all types of phospholipids, there are few functions, 10 functions that are represented here. The first function is they act as membrane structures with proteins and also allow the movement of nutrients through the permeability or the pores that are present in the cell membrane. Now, they also maintain in ETC structure in the mitochondria, the electron transport chain function of the mitochondria, that is, it is involved in ATP and ADP generations, and it maintains the structure of the mitochondria. It also helps in fat absorption in the intestine, and it is required for lipoprotein synthesis for lipid transport. And it prevents fatty livers by acting as a lipotropic factors, and the sources of arachidonic acid are involved in eicosanoid synthesis, like the prostaglandins and thromboxans. Their role of phospholipids is to reverse the cholesterol transport. So whatever the cholesterol that is forming, it is going to be reversed. And phospholipids also act as surfactants in the lungs, the dipalmitoyl lecithin, which is involved in smooth contraction and relaxation of the lungs. The cephalins are important in and participate in blood clotting. So they are involved in the blood clot formation. PI or the phosphatidyl inositol or IP3 and DAG are involved in the hormone signaling. So that covers the lipids. We have covered the definition of lipids, the classification of lipids. So we'll just go through classification before we end this video once again. So here is an image pictorial representation of a lipids. So these can be divided into various types. So one is a triglycerides. These have fats and oils at them. So fats and oils, fats are usually solid at room temperature, and liquids are oils are liquid at room temperature. The glycerols are 3 fatty acid chain groups, and they can also be fatty acid can also be called as saturated and unsaturated based upon the presence or absence of double bonds. So in saturated fatty acids there is no double bond. In unsaturated fatty acids there are double bonds. So saturated fatty acids are bad and unsaturated fatty acids are said to be good. So in saturated fatty acid like cheese and processed food are observed, and in unsaturated fatty acids they are obtained naturally. Phospholipids, so here I told it is present in the cell membrane. So in a cell, the outer ring structure consists of a cell phospholipid layer, the hydrophilic head water loving, or the outside region is hydrophilic head, and the inner one, which is hydrophobic tail, is water hating structure. So this is a phosphobilipid layer, and the water loving group is phosphate group, and the water hating group or hydrophobic group is a fatty acid because of the insolubility of lipids in water. So phosphate group is towards the water loving side, and the fatty acid is towards the inner side of the cell, thereby keeping the cell protective. Now the steroids are a type of fatty acids that are having 4 carbon based rings. So some of the steroids are cholesterol, testosterone, estrogen, vitamin D, and cortisone. These are also involved in the steroid synthesis. Now waxes are another type of simple lipids. So these are having long carbon chains and are usually solid at room temperature, and they don't have water and repel water. So waxes are seen in plants and human ear and bee wax as well. So these are some of the lipids. So the classification of lipids, there are 3 types as I told: simple lipids, compound lipids, and derived lipids. Why I am stressing on classification of lipids is because there are some chances of having 5-mark or 3-mark question based upon classification of lipids. They might ask the examples of simple lipids or compound lipids or phospholipids in the 3-mark questions. So knowing the names of certain lipids can be helpful while writing your exams. So taking an example of each type of lipid can give you the 2 marks. Now simple lipids are classified as fats and oils or waxes. Fats and oils are solid and liquid at room temperature. They have fatty acid plus and glycerol group. In waxes, you have fatty acid plus alcohol group. In compound lipids, you have the complex lipids are made up of fatty acids, a nitrogenous base sometimes, and the additional group, which can be phosphate group, carbohydrate group, or sulfur group, or a protein group that will combine to form a compound lipid. So the compound lipid can be divided as phospholipids, glycolipids, sulfolipids, and phospholipids can be further divided as lecithins, cephalins, plasmalogens, and sphingomyelin. I have explained in detail through the video, and here is just a glimpse of whatever we have read. And glycolipids have a carbohydrate group along with the lipids, and the examples are cerebrosides and gangliosides. Sulfolipids, as I told, they are a combination of sulfur group plus lipids. Now derived lipids are obtained by hydrolysis of the whatever simple and compound lipids are present. So they might have alcohol. On hydrolysis of a simple lipid, you might obtain alcohol. On hydrolysis of fats and oils, you'll get fatty acids, and hydrolysis of any of the components will give you sterols and isoprenoids as well, which provide terpenoids and carotenoids. So that covers the